So today we shall be discussing diabetic ketoacidosis, which is an important complication of type 1 diabetes. It can also develop in late type 2 diabetes, but basically it is associated with type 1. Type 1 diabetes is insulin dependent diabetes mellitus, and when there is severe insulin deficiency, though there is presence of glucose in the body, it cannot be metabolized because there is no insulin, and the body perceives it as a state of hypoglycemia. This relative hypoglycemia leads to production of hormones that increase the level of glucose in the body. These hormones are glucagon, growth hormone, epinephrine, and cortisol. So the effect of these hormones would be to increase the level of the glucose in the body. These hormones function by increasing gluconeogenesis and glycogenolysis. Also, these hormones break down lipids into free fatty acids. They cause lipolysis and increase in level of free fatty acids, which in the liver are converted into ketone bodies. These ketone bodies, as we know, there are three important types of ketone bodies: acetone, acetoacetate, and beta hydroxybutyrate. This acetone is responsible for the fruity breath that we find in DKA patients, and the beta hydroxybutyrate is responsible for the nausea, vomiting, and hence dehydration that is found as a sign in patients of DKA. Because of increase in levels of ketone, free fatty acids, and hence ketone bodies, these ketone bodies are actually acids, and they lead to a state of high anion gap metabolic acidosis. Now, as a reminder, what are the other causes of high anion gap metabolic acidosis? It can be remembered by a very common mnemonic called mud piles. M is for methanol poisoning, U is for uremia that happens in CKD, D is for DKA, the present scenario that we are discussing, E is for polyethyl glycol poisoning, I is for INH toxicity, L is for lactic acidosis, E is for ethanol poisoning, and S is for salicylic acid poisoning. So these are the common causes of high anion gap metabolic acidosis. Now, because of the state of acidosis, there is a increase in the amount of H plus inside the intravascular space. Now we know that the body compartment can be divided into intracellular fluid and the extracellular fluid. Again, extracellular fluid is interstitial space and intravascular space. We cannot measure the amount of substrates in the interstitial space, but we can definitely measure the amount of cations and anions in the intravascular space. In fact, the serum levels of electrolytes which we measure are actually the levels inside the intravascular space. So, in diabetic ketoacidosis, there is an increase in level of H plus in the intravascular space. And we know that in the intracellular fluid, there is potassium. Potassium is the major intracellular fluid cation. Now, the body tries to deal with this acidosis by shifting this potassium from the intravascular space into the intracellular space, and because there is a movement of potassium ion, because there is a movement of H plus ions inside the intracellular space, one positive ion moves in, one positive ion has to move out. So H plus moves in and potassium moves out into the intravascular space. So ultimately what happens, the intracellular space potassium level drops and the extracellular, that is intravascular space potassium level increases. This is measured as hyperkalemia, because remember the serum levels that we measure are the levels of cations or anions in the intravascular space. So ultimately what has happened, the body's potassium level has not increased, only that the potassium from the intracellular fluid has shifted to the intravascular space. So what has happened is the level of potassium inside the intracellular fluid has reduced, and that is not good for the body's homeostasis, because if we remember basic physiology, the resting membrane potential of any cell is maintained by potassium ion and the movement of potassium ions inside from inside to outside. This maintains a diffusion potential, and this creates a negative resting membrane potential inside the cells, and then these cells can be depolarized. Now, patients of DKA, because of this hyperkalemia, they present with weakness. Now, why does it happen? Let us draw this thing out again. This is intracellular space and this is intravascular space. So what has happened in DKA? The levels of potassium here has increased and here it is low. As we discussed in the previous slide, because of movement of potassium from the intracellular space to the intravascular space, the inside of the cells developed a negative resting membrane potential, and then they could be depolarized. Depolarization is basically the increase in the resting membrane potential from negative to positive. So what happens if the potassium level is high in the intravascular space? You can understand that less negative charge will be created here. So what does that mean? That means the negative resting membrane potential has actually fallen. The negative charge has fallen. That means the positive charge has increased. So this is a state of mild depolarization. Now we might think that because already some kind mild depolarization has happened, then action potential development would be easier, because only a little bit of increase in the in positive potential is now required. But this is not so. Though there is a mild depolarization, what happens because of this mild depolarization is that the inactivation gate. See, this is a sodium channel inside the cell. The sodium channel has two gates, activation gate and inactivation gate. When both of these are open, there is influx of sodium inside the cell, which causes action potential. Once the cell is mildly depolarized, this inactivation gate, it closes. Inactivation gate closes in cases of mild depolarization, and sodium cannot enter inside, and hence there is no action potential development. And there is no action potential development means that the muscle will not contract, and there is the classical finding of weakness in these patients. Now, so the main clinical features are fruity breath, as we already discussed, nausea, vomiting, dehydration because of that, and weakness. We have discussed the pathophysiology of each of this. Now, because as we have already discussed, there is a state of high anion gap metabolic acidosis. The body will respond to it by respiratory alkalosis. So to cause this respiratory alkalosis, we have to increase the respiratory rate so that carbon dioxide is lost and the acidosis is reduced. That is why these patients develop a rapid deep breathing, which is known as Kussmaul's breathing. The treatment of diabetic ketoacidosis is institution of insulin which is absent, giving IV fluids and correcting the potassium levels. So hope this clears up diabetic ketoacidosis for you.